Here we have question number 106. Question says, a cell in a quiescent stage is. Students, just to give you an idea, please remember that though a cell has got two more important phases which we keep on talking all the time, one is I phase, that is interphase, and other is M phase. We should not forget a very important phase that goes by the name G0 phase. Yes, I'm talking here about the very distinct G0 phase. Now, this phase is distinct because here cells will not divide. They will not proliferate. Okay? But they are pretty much alive. They are not dead. So metabolically active they are. So let's see what are the options given here. The options here say, number one, cell in quiescent stage is non-proliferating. It is absolutely correct. Cells do not divide here. Then it says metabolically inactive. That's incorrect because they are metabolically active. They are healthy. They are living. They're not dead. Actively dividing. No, they do not divide. Last, a meristematic cell in plants. That's not going to happen because meristematic cells are actively dividing cells. They are actively dividing cells. Therefore, students, what is the right answer? Yes, option number 1 is the right answer. Alright, let's move further. Next, we have a diagram-based question. Let's see what is this. The question says, identify the stage of cell division and select what is true for it. Students, we have to see what is true. So if you go back to your notes, you will see that this diagram is the diagram of anaphase. A phase where what happens? Where chromatids separate. Now let's see what is given here. Option number 1 says microtubules of spindle fibers get attached to the kinetochores. Is that true? Let's see other statements also. Best stage to study the shape of chromosome. I'm totally happy with this statement. Why, students? The reason is that in anaphase you see the shapes of chromosome, whether it is I shape, whether it is V shape, whether it is J shape, whether it is L shapes. So we can see those acrocentric, telocentric, if you remember that. So yes, in anaphase we are able to see the shape of chromosome because here what happens? Here centromere splits and chromatids separate. Then nuclear envelope reassembles around the chromatin cluster. That's not right. It marks the end of karyokinesis. Not correct. Therefore, we already have chosen the right answer will be option number 2. Please remember, in anaphase there is splitting of centromere. There is splitting of centromere and separation of sister chromatids, which is why we get a better visibility of the shapes of chromosome. Next we have is question number 108, which is match the following type. It says match column 1 with 2 and then select the correct option. So if we see here, we are given zygotine, dikinesis, picatinine and diplotin. If you remember, these all are the stages of prophase 1. You remember that? So the stages were leptotine, number 1. Then is zygotine, picatinine, diplotin and dikinesis. So in order to remember this, some students have got different mnemonics. Some of you have different mnemonics. For you, I can give you one tiny mnemonic. Lazy zebra pulls dead donkeys. Just to remember that, lazy zebra pulls dead donkeys. Okay, just to remember. Then let's see what is happening in all of these phases. Students, if I talk about zygotine, it is the phase where pairing of homologous chromosomes do occur. Then talking about dikinesis, which is at the end, it is marked by terminalization of chiasmata. Chiasmata terminalizes. Then we have pecitine, which is the third stage. What happens in the pecitine? There is formation of recombination nodule. Then in diplotin, we can see desynapsis happens. So if we see connecting all these functions with the choices given here, we arrive A has 2, B has 4, C has 1, D has 3, which is option number 4. So I'll write down the answer is option number 4. Now let us move further. Next we have is 109. Question says, which of the following is a mitogen? What is the meaning of a mitogen? Mitogen is a substance or a compound which induces mitosis. And insulin is one of those. So we'll write down that the answer is option number 4. Moving further, next question is a very interesting question and my personal favorite too. The question says, male gamete of a plant has got 10 picogram of DNA. What would be the amount of DNA in the meiocyte of the same plant at its G2 phase of the cell cycle? Whenever such kind of a question comes in your exam, quickly draw a cell cycle, which will help you. So here I have a cell cycle and I will make this as the M phase. And then what do we have? We have this, number 1 is G1, then we have is S, then we have is G2. We remember that G1 plus S plus G2, all of them are the part of interphase represented by I. So a mother cell has to begin its journey, right? A mother cell will begin its journey at G1 phase. In S phase, it will duplicate its genetic content. Also in G2, the genetic content will still remain duplicated. And in the M phase, it will decide to undergo either mitosis or meiosis. Now remember, the question says, so here students, in G1 phase, we don't know what is given. In S phase, we don't know what is given. In G2, we don't know. But what is given? Male gamete. That means one fine day, this mother cell will undergo into M phase, and M phase here has to be meiosis because male gametes will be formed only by meiosis. This means that this mother cell will form 4 male gametes because one meiosis leads to the formation of 4 haploid cells. Okay. Now it says that one haploid cell, one male gamete, which is a haploid cell, has 10 picogram of DNA. So this cell has got 10 picogram of DNA, which means the rest of the other 3 haploids will also have 10 picograms of DNA. So here also we'll have 10 picogram. Here also I have 10 picogram. Here also I have 10 picogram, which means this is in total what? 40 picogram. So this 40 picogram was present here in the mother cell at G2 phase. That means in S phase it might have duplicated up to 40, which means in G1 the DNA was only 20 picograms. G1 it has 20 picograms. S phase duplicated its content. We know S phase is synthesis phase where replication of DNA will occur. So 20 to 40. In G2 again it will still remain 40, and then after G2 this mother cell decides that I will undergo meiosis and I will create 4 haploid cells. So this 40 is going to get distributed in 4 haploid cells. Now what is the question asking us? I think now we have arrived correctly. So the question says male gamete has 10 picogram, correct? What is the amount of DNA in the meiocyte? Meiocyte, the mother cell, which will undergo meiosis of the same plant cell at its G2 phase. So what is in the G2 phase? 40 picograms is present in the G2 phase. Correct? So students, what is the right answer? We all have learned that answer is option number 3 with 40 picograms of DNA. I hope that was easy. Now let's take another question.